Good day, grade 12 learners. Welcome to today's Life Sciences Paper 1 revision lesson. My name is Fauzi Rahman, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Right, my colleague Radin Kruger and I are going to take you through this afternoon's lesson on Paper 1 revision. Now, with Paper 1, let's look at the weightings, the topic, and the mark scheme. If you look at human reproduction, responding to the environment, and endocrine and homeostasis in humans, those are quite large amounts of percentages and marks. And responding to the environment in humans is 54 marks. That's like 1/3 of the paper. So make sure you study these sections well, and we are going to go through all of them, including plant hormones. The structure of your paper is 150 marks for 150 minutes. That means in life sciences, we are 1 mark per minute. So you need to keep to time. Section A is 50 marks. Section B is 100 marks, made up of 2 questions, question 2 and question 3. So I would suggest that you try and go through enough section A's as possible so that you are quick through section A, and then by 9:30 you should at least then be finishing off and starting your question 2 so that you have enough time for the higher order questions in question 2 and 3 so that you can get good marks. The exam guidelines for human reproduction shows us that we need to study the female reproductive system, the male reproductive system, puberty, menstrual cycle, hormonal control, and development of the fetus. The diagrams that you could be asked to draw in this section is the diagram of the ovum, so make sure you practice that, and the diagram of the sperm cell. Please practice this. Make sure your diagrams have a title and you label your diagrams. So we're going to begin with the male reproductive system. It consists of a pair of testes in the scrotum. And remember, the scrotum is outside the body because sperm is sensitive to temperature. And the epididymis, and sperm is also manufactured or produced in the testes. In the epididymis, it's where it is stored until it matures. And then we have the vas deferens or the sperm duct. And then as you continue along the same line, it becomes the ejaculatory duct, and the same duct, if you follow through, becomes the urethra. This is the passage of semen. Now remember, semen is made up of sperm and fluid. Then we have the accessory glands, the seminal vesicle. This is the seminal vesicle. And we have the prostate gland here, and then Cowper's gland, and then the penis. Now, the seminal vesicles, they produce seminal fluid to transport sperm cells and fructose for nourishment of the sperm. Then we have the prostate gland that secretes a milky, slightly alkaline fluid that neutralizes the acidity of the vagina, and it has enzymes and nutrients for the sperm. Cowper's gland secretes fluid for motility of the sperm, and the secretions of the prostate and seminal vesicles nourish the sperm, enabling them to swim, and they also provide protection for the sperm. Remember, it's the tiniest cell and it needs nourishment and protection. It's very sensitive. We're going to look at an activity. So it says, give the letters of all the structures that are involved in the following: the production of fluid, the fluid part of the sperm, of semen. So remember, semen is made up of sperm and fluid. Now we are asking for the fluid part of the semen. That means we're not counting sperm. So it should be the glands that are producing the fluid that then that will be part of your answer. So that will be now your A, your seminal vesicle there, your prostate gland, and your Cowper's gland. That's A, B, and C. transportation passageway of sperm and semen. So now we're incorporating sperm as well as semen. So just be careful when you are answering your questions. And this is your transport for your sperm and your semen. That's E. But remember, sperm cells come from the epididymis, so it'll have to include the vas deferens here, which will be D and E. Let's see if you get that right. Okay, and gland B, we've already discussed, that's your prostate gland. Now, the hormone that is produced by the testes that brings about the development of male secondary sexual characteristics, which you should know, is testosterone. Test results show that this man has a low sperm count. Explain why a doctor would advise him against wearing tight underwear. Now, remember, firstly, that the scrotum, the testes are outside the body, and that is because of temperature. The body temperature is too hot and sperm cells are too sensitive. So there must be a reason against wearing tight underwear, because tight underwear will be too close, will bring the testes too close to the body, and that will cause the temperature of the testes to go up, and it will be too high, and sperm will not mature. The production will be negatively affected. Okay, so if you say sperm cells will be affected, or anything in life sciences will be affected, you got to say how. Negatively affected. So then the sperm will not be produced properly, and that's the answer that we want. What would happen if to male's ability to reproduce if part label D is blocked off? Part label D, we said, is the vas deferens. If it is blocked off, cut off, anything to that effect, no sperms will be transported to the penis, which will result in no fertilization because there's no sperm, and no offspring will be produced, and no reproduction will be able to take place. Now we look at the female reproductive system. It is made up of your birth canal, right, your vagina, the mouth of the uterus, which is the cervix. What lines the uterus is the endometrium, or the lining of the uterus, or the uterine lining, but we will say endometrium. And then we have the fallopian tubes and the ovaries that are attached by ligaments there, and the fimbri, or the cilia over the ovary from the fallopian tube that helps to move the ovum along the fallopian tube. So it consists of a pair of ovaries, a fallopian tube, the uterus, the vagina or the birth canal, and the external opening. The fallopian tube is to transport the ovum to the uterus. The uterus is for protection of whatever is developing inside, and the birth canal we spoke there is the vagina. Right? And your ovary, remember, sex hormones are produced in your ovary. So because it produces a secretion, then it's a gland. Right, let's look at a question. Identify part A. Now, if you look at part A, it is the fallopian tube. C then would be, we spoke about this, the cervix, or the opening to the uterus. Give the letter only of the part where the following takes place: meiosis. Now remember, meiosis takes place in a specialized gland we spoke about, and that is B, which is your ovary. But because it says letter only, you will say B. And where fertilization takes place, that will be your fallopian tube. Therefore, that answer is A. Two functions of part E. This is part E, it's your uterus, right? That is for the developing embryo or the fetus, or as it grows on to be the baby. It's responsible for the protection of the embryo from implantation to birth, and provides space for the developing fetus. Name the glands which secrete nutrient-rich fluid for the sperm to reach part A. Now we're talking about the sperm cells to reach part A. So we want nutrients for the sperm, and those are the seminal vesicles. We're going to take a look at puberty. Puberty refers to the age at which sexual maturity occurs and sex hormones are produced. And this leads to the secondary sexual characteristics. So when you are born, you're either a male or a female. Then later on, once you reach puberty, your body changes, and that is because of your sex hormones, and secondary sexual characteristics form. So in the male, it's caused by testosterone, and this starts when the pituitary gland secretes the follicle stimulating hormone, which stimulates the testes to produce sperm. Then the luteinizing hormone stimulates the testes to produce testosterone. And some of your secondary sexual characteristics that are caused by testosterone will be hair growth on the face, more muscular physique, enlarging sex organs. And if we look at for the female, the pituitary glands also secrete the follicle stimulating hormone, which stimulates follicles in the ovary to secrete estrogen. And then the luteinizing hormone stimulates the development of the corpus luteum in the ovary to produce progesterone. Now remember, that's not responsible for secondary sexual characteristics. It's estrogen that causes the secondary sexual characteristics. And what happens at puberty, then underarm hair develops, breasts develop, and widening of the hips, and it separates girls from women. Okay, now we're going to look at an important process that occurs at the start of puberty, and that is gametogenesis. In males, it is called spermatogenesis. Now, the production of sperm, you need to learn how to draw this diagram. So we will go through this quickly. You can see here that there is the head portion of the sperm, there is a neck region, and a long tail. Now, remember that I said you need to be able to draw this diagram. So make sure the head is in proportion to the tail, and you have a distinguished head, neck, and a tail region. The shape of the head is important, and it must be torpedo shaped. We don't want circular heads or very triangular. Right at the tip is the acrosome that has enzymes to digest the wall for fertilization, and then we have the nucleus, which contains 23 chromosomes. So it has 22 autosomes and 1 gonosome. That gonosome could either be an X or a Y, because remember, the number of chromosomes in the nucleus is haploid, so there's only one set of chromosomes. Then we have the neck region, which has a large amount of mitochondria, and that is to provide energy for swimming. And then we have your tail, which enables the sperm to swim fast, okay, for motility, to be able to allow the sperm to swim. Now, the process of spermatogenesis is exactly as it is in your exam guidelines. Under the influence of testosterone, diploid germinal epithelial cells in the seminiferous tubules of the testes, they undergo meiosis to form 4 haploid sperm cells. So these, make sure you learn and understand this. Now we look at gametogenesis in females, which is called oogenesis. Here we have the nucleus that contains 22 autosomes and 1 gonosome. Now remember, the gonosome for the female can only be X. X and X is a female, so we can only produce X gonosomes. There's a cytoplasm for nourishing the ovum. There's a cell membrane that allows one sperm only to penetrate, and a jelly-like layer to protect the ovum against drying out. Now, if we look at oogenesis in females, remember there's diploid germinal epithelial tissue lining. Just as it was lining the testes for males, it lines the ovaries for females. And then these cells undergo mitosis to form many follicles, right? They undergo mitosis, they form many follicles. At the start of puberty and under the influence of the follicle stimulating hormone, which we will abbreviate as FSH, one cell in the follicle grows and undergoes meiosis to form a haploid functional ovum. Okay, the diagram, so we've done one on the sperm, so I'm going to move on then to look at the next question. Okay, we're going to look at the menstrual cycle here. The menstrual cycle is made up of 2 cycles. It is made up of the ovarian cycle and the uterine cycle. So if you look at this diagram, if you understand this diagram well, you can apply this to any diagram you are given in the exam. And remember, you will not get this diagram. They will probably give you two hormones only, graphs. You could get a pie chart. If you understand what happens from day 1 to day 5, from day 7 to day 13, what happens on day 14 and thereafter on day 28, then you'll be able to apply that knowledge to any diagram that you are given. So this is a very important diagram for you to learn and to understand. So if we look at the menstrual cycle, you can see here this part here occurs in the ovaries because it's the follicles that are developing. Then we have all the hormones here, and we have the uterine cycle at the bottom here, which shows the endometrium and what is happening in the uterus. So there's various places that these processes are occurring. As we said, the ovarian cycle is occurring in the ovaries. Then we look at these graphs. Now, FSH and LH, these two, remember they are released by the pituitary glands. So they are the pituitary hormones. Then you've got to differentiate between the ovarian hormones. The ovarian hormones are produced in the ovaries. Remember I said the ovaries are glands because they produce hormones, and those are estrogen and progesterone. And then there's a diagram here of the uterine wall or the endometrium, which is the word we are looking for. Right, that's your y-axis. If we look down on your x-axis here, we can see there's days. So if you look at the days, you've got day 1, day 7, day 14, and day 28. Now you've unpacked this diagram. Let's look at what is happening. We're going to use this mnemonic called FOLP, and you need to learn this, right? So you start off with FSH, because remember we said during puberty, FSH is released by the pituitary gland. Now, when that hormone is released, it causes follicles in the ovaries to start developing. So there's the follicles. These are the developing follicles that are forming in the ovary. Now, what do? They produce the hormone estrogen. So if you look at the red graph here, the red line, you can see that as the follicle is developing, it's producing more estrogen. That's what the follicles do. They produce estrogen. Now remember, in this one follicle that is developing, it's forming an ovum, right? So one ovum is developing and maturing and growing by the process of mitosis. So now, when estrogen is forming, let me go to the function of estrogen. What does estrogen do? Guys, this is extremely important for you to understand that estrogen builds the endometrium. It makes it thick, it makes it vascular, it makes it glandular. What do I mean by vascular? The blood vessels are filling in the endometrium during this time. Mucus is filling in the glands, and you can see here the endometrium is becoming thick. Right. Now, when we reach this part here, this Graafian follicle, the ovum is matured, and this Graafian follicle produces the largest amount of estrogen. You can see here the estrogen is quite high. The level of estrogen has reached its peak, and then this triggers the pituitary gland to produce the luteinizing hormone. Now you can look at the orange graph and you'll see it spikes on day 14. What happens on day 14 is ovulation. What causes ovulation is the high amount of estrogen that triggers the pituitary gland to release the luteinizing hormone. What does the luteinizing hormone do? It causes ovulation to occur, and the second thing it does is causes the corpus luteum to form. So, when the process of ovulation occurs, what is ovulation? It is expelling the ovum from the Graafian follicle. That is ovulation. So, there's your ovum and your Graafian follicle. Now, your Graafian follicle is an empty Graafian follicle because the ovum has been released. So, the luteinizing hormone now causes this empty Graafian follicle to become the corpus luteum. What does corpus luteum do? So we spoke about the FSH, we've spoken about estrogen, we've spoken about the luteinizing hormone, and now we're going to speak about progesterone. So the corpus luteum produces the hormone progesterone. What does progesterone do? Progesterone further increases the thickness of the endometrium. So you can see here the blue line, the blue graph. As progesterone is increasing here, you can see that the endometrium is increasing. So when progesterone is at its highest point, the endometrium is at its thickest. So progesterone further increases the endometrium, making it more thick, more vascular, more glandular. Guys, we want you to say those words more, because remember, estrogen thickens the endometrium, makes it vascular and glandular, but progesterone makes it more thick, more vascular, more glandular, right? Thereby maintaining pregnancy should there be a fertilization and an implantation occurring. So you cannot say P for pregnancy, P for progesterone, it maintains pregnancy. No, we want you to tell us how it maintains pregnancy. So you got to talk about the endometrium being vascular, more glandular, and more thick. Right. So now, when the corpus luteum forms, we said the progesterone forms, and you can see that the endometrium is quite high. Now, the corpus luteum, can you see here, it's getting smaller and smaller. It's disintegrating or degenerating, which means that no fertilization took place. So what happens? The progesterone level immediately drops down. And when that happens, you can see how the endometrium is going down here as well. And that would lead to this process that will start again. From day 28, we'll move back to day 1, and that process will be menstruation. So remember, I said the menstrual cycle is made up of the ovarian cycle and the uterine cycle. I also want to say that the menstrual cycle is a 28-day cycle. Menstruation is only a process in the menstrual cycle, and that process lasts up to approximately 5 to 7 days. So if you look at the days here, remember day 14, around day 14 should be ovulation, and looking at what happens with regards to the ovary and your hormones, you must be able to apply this to any graph that you are given in the exam. If they say it is pituitary hormones, then remember it's going to be FSH and LH. If they talk about ovarian hormones, it's going to be progesterone and estrogen. So take a highlighter, highlight, underline what they are giving you in the exam. I think I've covered everything there. Okay, I just want to speak about one more thing, and that is the negative feedback mechanism. So if you look at the green line here, right, you can see that as the green line increases, the green line is FSH, the follicle stimulating hormone secreted by the pituitary gland. Now, the function of this hormone, FSH, is to produce follicles, and that is eventually to produce a functional ovum. But the negative feedback mechanism is when FSH is starting to increase or increases, it inhibits, it prevents progesterone from forming. Right? So progesterone is low while FSH is high. Now you can see here progesterone is low while FSH is increasing, and the moment progesterone increases, the blue graph, progesterone is increasing, FSH drops. FSH is now low. Okay? So the thinking behind this, or the reasoning, is because FSH produces follicles to produce a functional ovum, as I said. And now, when progesterone is high, there could be a potential pregnancy. So there's no need for the body to waste energy and produce more follicles. The follicles will form when the progesterone level drops, which means that there's no fertilization, there's no pregnancy. We need to start this menstrual cycle, this process all over again, and that's when FSH will start increasing to produce more follicles. So it's a negative feedback mechanism because a high amount of progesterone. Guys, you've got to say a high amount of progesterone. Remember, we have all these hormones in our bodies at all times, but at certain time there is more of a hormone than the other hormone. So we got these hormones in our body, but at the time of ovulation, for example, there's more LH. When we are menstruating, then we have to produce more FSH so the follicle can form, and at that time there is you are menstruating, there's no pregnancy that can occur, so progesterone is low. So our body saves energy by producing certain hormones at certain times. So you got to say a high amount of progesterone inhibits FSH, and vice versa. A high amount of FSH also inhibits progesterone. Okay, right, let's look at the second graph. Now, if fertilization takes place, you can look at the corpus luteum here. It doesn't disintegrate. It's not degenerating. It's not getting smaller. Look at the progesterone levels. This blue line here. This progesterone is not decreasing. It's remaining high. Look at the endometrium. It's remaining thick. Therefore, there was a fertilization that has occurred. So that is how you will know if fertilization occurs, because this endometrium will remain thick, progesterone levels will remain high, and the corpus luteum will remain big. It will not disintegrate or degenerate. And then if you look at an interesting graph here, that is body temperature, you can see at the time of ovulation, your body temperature increases. So you will know when you are ovulating, when your body temperature increases slightly and drops after that. Okay, let's move on. Now, if there was a fertilization as we saw in the previous slide, what happens is the first, okay, let's look at this. There's your ovary and it releases an ovum, and this ovum is traveling along the fallopian tube. Then sperm is inserted here into the vagina, and the sperm travels along the fallopian tube. By chemotaxis, the sperm will sense where the, by chemicals, there's a reaction, it can sense where the ovum is, it will swim towards the ovum, and one sperm the acrosome will dissolve that membrane, that sperm will enter this ovum, and fertilization is the fusion of a haploid nucleus of the sperm with the haploid nucleus of the ovum, and this process occurs here in the fallopian tube. So you can see here, this is enlarged in that diagram there. Now, the first structure that forms after this process is this structure here called a zygote. Now remember, the sperm is haploid and the ovum is haploid. So the structure that forms is a diploid zygote, 2N. Remember, now it has a complement from paternal chromosomes, father, and maternal from the mother. And now we have a diploid zygote that undergoes mitosis now to grow, okay, until it forms a morula, which is a ball, a solid ball of cells. And then we have a blastula. The blastula, remember the spelling, and this is a hollow ball of cells. So this ball has about 64 cells and there's fluid inside, okay? And by the time it reaches the zygote forms the blastula, it's still traveling along the fallopian tube here. And then it's a blastula, and it embeds itself or attaches itself to the endometrium in this diagram here, which is enlarged here. This process where the blastula attaches itself is called to the endometrium is called implantation. So if a fertilization occurs, but implantation does not occur, there will not be a pregnancy. So this implantation process is important for a pregnancy to occur. Now we're talking about the development of the fetus in the uterus. Fertilization has occurred. So let's look now at the development of the fetus. Now this structure here is called chorionic villi. It's finger-like projections of the chorion, which is a structure here. The outermost structure here is a chorion. The inner one here is the amnion, which produces the amniotic fluid. This is the amniotic sac. Here we have the umbilical cord, and the chorionic villi is this one here. So these finger-like projections of the chorion, they project into blood spaces in the uterus of the mother. They provide maximum contact with maternal blood together with the endometrium, or the uterine wall, together with the uterine wall and the chorionic villi. It forms a structure here called the placenta. Now, the chorionic villi and the mother's blood, the sinuses here, they come close together, but they don't touch, they don't mix, right? Diffusion occurs between them. So the blood of the mother and the blood of the fetus never ever mix, although they're coming quite close to each other, close proximity to each other, but they never mix. Now, the umbilical cord is a flexible cord containing 2 umbilical arteries and 1 vein. Remember, the arteries carry carbon dioxide from the fetus to the mother, to the placenta to be discarded, and the umbilical vein brings nutrients from the mother to the fetus to grow. Then if you look at the amnion, it provides or secretes amniotic fluid that protects the developing fetus. Amniotic fluid is full amniotic fluid inside here, and that acts as a shock absorber and protects the growing fetus against temperature changes, against dehydration and desiccation. Then we have the placenta, which allows for gaseous exchange between the mother and the fetus, providing nutrients to the fetus. And another important function of the placenta is once the approximately 3 months or about 3 months into the pregnancy, the corpus luteum will now disintegrate, and the placenta now takes over the job of producing the hormone progesterone. So this is very important that the placenta now produces the hormone progesterone. And remember, we need progesterone to maintain the thickness of the endometrium, to keep it thick, vascular, glandular, so that it can maintain that pregnancy. Okay, let's look at an activity quickly. The diagram alongside shows hormone levels in the female menstrual cycle. Identify pituitary hormone X. So we're telling you it's a pituitary hormone, right? And you can see that it's spiking at day 14. So that must be LH. Then we look at the ovarian hormone Y. We're saying it's ovarian, and you can see that it is increasing after day 14. So it must be coming from the corpus luteum. Therefore, it must be progesterone. Name the hormone responsible for thickening of the endometrium before day 14. And that has to be the hormone that builds the endometrium, remember? So that is estrogen. Explain the effect on reproduction if a female is unable to produce hormone X. So if she is not able to produce the luteinizing hormone, the follicle will not be stimulated, no ova will be released, ovulation will not occur, and therefore this female will not be able to reproduce, and obviously fertilization will not occur. Explain with evidence from the graph. It means you need to use the graph which suggests that fertilization did not take place. We go and look for progesterone. So let's look at ovarian hormone Y, and you can see it's decreasing, which means that no fertilization took place. The corpus luteum has now disintegrated. Right, and you can see hormone Y or progesterone level starts to decrease due to the corpus luteum disintegrating. Right, I want to look at another activity quickly. This diagram represents different stages in the development of the lining in the uterus during the menstrual cycle. So you need to unpack any data that's given to you, any diagram or any data. You can see here days of the month, and there's a uterus here, and you can see there part A is the cervix is there, and this is the vagina, and you can see that something is coming out here, and then that is probably blood due to menstruation. And then you can see here the thickening of the uterus. So that is the endometrium becoming thicker as we're moving along here. So identify part A, which we discussed. It's the vagina. The process that's occurring in these first few days, from day 1 to approximately day 5, the shedding of the endometrium, that's menstruation. And then the ovarian hormone that is at a higher level from day 15 to day 28, and that is going to be progesterone. Remember to make the endometrium more thick. Explain the thickening of the uterine lining from day 7 to 13. So the Graafian follicle secretes the hormone estrogen, which builds the endometrium, making it thick, vascular, and glandular. Right, contraceptive pills, some of them have a high dose of progesterone. Explain why a woman using this kind of pill will not fall pregnant. What does progesterone do? The high levels of progesterone in the blood inhibits FSH. Remember, we said the negative feedback mechanism by the pituitary gland. No follicle will develop to form the Graafian follicle. No ovulation and no fertilization will take place. So a contraception that is used like the pill has a high amount of progesterone for this reason. Uterine fibroids are small, non-cancerous lumps that can form outside or inside the uterus. They cause pain, bloating, and abnormal menstrual periods. Explain how uterine fibroids can affect fertility. So if you are trying to conceive or have a baby, but you have fibroids, then what happens is the uterus could be blocked by the fibroids, thus preventing fertilization, or it could be blocking the uterine lining, preventing implantation of the embryo. Remember, we said if there's no implantation, there's no pregnancy, or growth and position of the baby will be prevented, and the fetus will not be able to grow properly, and that could result in a miscarriage. Right. I'm going to look at the next slide because we are pressed for a bit of time. I'm going to move on to plant hormones, okay? I'm going to look at plant hormones. So this is a section that also has a bit of marks that you generally learners do quite poorly in. So let's please pay attention. Right, what does the exam guidelines say? The functions of the plant hormones, you need to know about auxins, gibberellins, and abscisic acid. Okay, those are the hormones that you need to know. You need to know the role of auxins in geotropism and phototropism, and the role of the hormones, of the plant as defense mechanisms, chemicals, and thorns. This section is 13 marks. Now we're going to look at phototropism and geotropism. A tropism is a growth response which makes a plant act in respect to a directional stimulus. So if you look here, the plant is growing upwards towards sunlight, phototropism, downwards here, the root, gravity, geotropism, right? There's light. When there is light, the auxins are damaged, so they move away from, they get damaged by sunlight. They do not like sunlight, they move away to the sunlight, and they stimulate, auxins stimulate growth in the shoot. So what happens is, there's your shoot, there's the auxins that are on the side, and what happens is it's stimulating growth. Therefore, this part is going to grow much faster than this side. And as it's growing faster, this side is trying to catch up, and it bends towards the light. If you look at the shoot, this is what it's explaining here. It moves down due to gravity, and because it's stimulating growth here, it's growing faster, and this side it's trying to catch up. Therefore, it's going to move upwards. And I'm going to look at the root quickly. Auxins inhibit growth in the root, and there's your auxins. So this top part is going to grow faster than the bottom, and therefore it's going to join the bottom, and due to gravity, this root is going to move downwards. We are just going to take a minute break and be back.
I want you to ask yourself these 3 questions. Number 1, what do I want? Do I want to become an A student, or do I want to become an average student? Number 2, I want you to ask yourself, how do you plan on getting there? Are you going to put more extra hours, or are you going to just sleep? Number 3, who will be with you in this journey? Because it is important that we associate ourselves with students who have the same vision as us, who want to become a student, who want to put in the extra work, who want to become better at what they do. Unsuccessful people do not plan. They just let life happen to them. But successful people are planning on how they want their lives to be. And right now, you plan on how you want your results at the end of the year to be like.
Sorry for the technical glitch. We're now moving on to phototropism and geotropism. Remember, we spoke about the plant hormones, auxins. Now, phototropism is when the plant grows towards the light, and geotropism is it's growing downwards towards gravity. When there is light, auxins move away from the light. So they're moving, if light is shining on the stem on this side, the auxins are going to move to the other side here. Now, what do auxins do? They stimulate growth in the shoot. So this part of the shoot is going of the stem is going to grow much faster because there's a lot of auxins. There's a high concentration of auxins on this side. So as the shoot grows faster, it's trying to catch up with this side that has no auxins, and therefore it's going to bend towards the light. And there's the shoot here. So this is also a shoot, and if the shoot is horizontal, then the auxins are going to go down because of gravity. So there's no light here, right? We are looking at gravity. The auxins are going to go down. And remember, because it's a shoot, the auxins are going to stimulate growth. So this bottom part is going to grow much faster than the top. And the top is going to try and meet the bottom, and that is going to grow upwards. When we look at auxins in the root, auxins, there's your root, your horizontal root. Auxins are going to move downwards due to gravity. Auxins inhibit growth in the root. So it's different to stems. In stems, it promotes growth. In roots, it inhibits growth. Please understand that differentiation. Now, this part here where the auxins are at the bottom of the root is going to grow much slower than the top, because remember, auxins are inhibiting the growth. So it's going to grow slower, and this one is growing much faster. Therefore, it's going to push it down and make this root grow downwards towards gravity. Right. The role of auxins in phototropism, when a stem is exposed to unilateral light, that's light from one side, the auxin concentration will be high on the dark side because light destroys auxins. More growth occurs on the dark side because auxins stimulate growth on the dark side. The stem bends towards the light. When the root is placed horizontally, on one side exposed to gravity, the auxin concentration will be high on the lower side. And remember, gravity attracts auxins, but also more growth occurs on the upper side because remember, auxins inhibit growth in the root. Therefore, the root bends downwards. Right, I'm going to answer this question. I'm going to look for the 2023 paper, and we're going to answer that one. But you have this presentation. Make sure your teachers send this to you, and you have the worksheet so you can do this, right? I'm going to just look at 2023. Right, this activity was from Mpumalanga, prelim paper 1. Seeds produce auxins development for fruit needed for fruit development. The function of these auxins during this stage has been investigated in different fruit. One of the investigations was done in strawberries. Since strawberry carries seeds on the outer surface of the fruit, these seeds can be removed without damaging a plant. A scientist did an investigation to determine if auxins are needed for fruit development. When you come across to determine, to find out, that is the aim of the investigation. That is where you're going to get your dependent and your independent variables from. So several strawberry plants started to form fruit, which was used 5 days after pollination took place. The scientists, she divided them into groups A, B, and C, as you can see here. Group A grew naturally. Group B was where she removed the seeds, and group C was still attached to the plant, but auxins, an auxin containing solution was covered this plant here. All the strawberries were left to grow naturally and harvested on day 25. So you can see here, strawberries harvested on day 25. Identify the dependent variable, which you got from the aim, which is development of fruit. One factor that was kept constant during this investigation, the same species of strawberries were used, and they were the same time period, 5 days after fertilization. Explain the purpose of A. Remember, A was the control because it grew naturally, and we need the control to compare. Give one way in which the scientists ensured reliability of the results. The seeds were removed and they did not grow, and here the plant seeds were left untreated and fruit developed there. Okay, I'm going to look at your other hormones that you need to know besides auxins. So as I said, you have this. You will look at the presentation and the worksheet. The other two hormones are gibberellins and abscisic acid. What is gibberellins or gibberellins for? Stem elongation, the germination of seeds, and it promotes flowering. Okay, you can see there. And abscisic acid is an inhibitor of growth, causes plants to become dormant during winter. Lack of water causes stomata to close when the plant wilts. So that's trying to protect that plant. So Human response to the environment is up next with my colleague Radine Kruger. You can have a quick comfort break, but please come back as quick as possible for the next important section. And remember, these sections also have a large amount of marks in your paper. So stay where you are. Goodbye and thank you.
Excellent. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you're gifted in, whether it's sports, it's academic, ucula, whatever that gift is, strive for excellence. Excellent. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you're gifted in, whether it's sports, it's academic, ucula, whatever that gift is, strive for excellence.
Good afternoon, grade 12s. Welcome back to the Life Sciences Paper 1 revision lesson. My name's Radine Kruger, and I'm going to go through human responses to the environment, as well as the endocrine system and homeostasis. Okay, so let's have a look and see what does the exam guidelines say. Right, so for the nervous system, we need to know the brain. We need to know the structure and functions of the following parts. So you need to go through those and just know basic where they are and a very basic function. The next thing we need to know is the spinal cord. And remember, the brain and the spinal cord together make the central nervous system. So we have a look at the structure of the spinal cord. And then you must also be able to describe a reflex action, possibly using a diagram, or they might give you a diagram. Then the next part that you need to know is the neurons, and know that they are specialized nerve cells, and we look at them in the context of how they function. So there's 3 different neurons. We're going to have a look at that, and then we're also going to have a look at the human eye. What's important there is the basic structure and the function of the different parts, as well as we're going to look at 2 of important functions in the eye, and that is accommodation and pupillary reflex. And then we're also going to have a look at the next part, which is the ear, the human ear. We're going to have a look at the structure again and the functioning of the different parts, the process of hearing, balance, and then some hearing defects. Okay, so let's have a look. And then remember, this topic, responding to the environment, is 54 marks. So it's a third of your question paper. So a very important topic, but there are certain aspects that you can pick out of there that you need to know. All right. So if we have a look at the nervous system, first of all, it's divided into different parts. And the first part we call the central nervous system there. And remember, the central nervous system, or sometimes we abbreviate it as CNS, is divided into 2 parts. And that is the brain and the spinal cord that runs down from the brain. So that's the central nervous system. Then the other part of the nervous system is the peripheral nervous system. And remember, periphery means the outside, the boundary. So the peripheral nervous system is all those nerves that branch off the central nervous system. Okay, so we find sensory pathways. Remember, there we have sense organs in our body, and they then are going to be stimulated by stimuli in the environment, things that are happening, changes that take place in the environment. That information is then carried via the sensory neurons or pathways to the central nervous system, and then some kind of response will be affected via the motor pathway. Okay, so and then if we have a look at our motor pathways, we've got the somatic, which is just regular body functioning, and voluntary, so where I can think about, I want to walk, I want to move my arm, I want to do whatever, talk. Then there is also the autonomic branch, and that is the part that controls things automatically. So I don't have to think about it. So while I'm busy talking to you, I don't have to think about, oh gosh, I better take a breath. Right, so it's happening automatically. Okay, and my heart keeps beating. All right, so, and then your autonomic nervous system is divided into two parts as well, and that is known as the sympathetic division, or the sympathetic nervous system, and the parasympathetic. Okay, and remember they work opposite, they are antagonistic. So whatever the one does, the other one does the opposite. All right, so let's have a look at the basic structure. So as I said, our central nervous system consists of the brain and the spinal cord. So let's have a look at some of the main parts. Some of the main parts. Let's go forward of the brain, and remember the front part here. Here is known as the cerebrum, and remember that is where our intelligence, our thought processes, those kind of things are taking place. Then we also have the corpus callosum, because our brain consists of two hemispheres, but they must be joined, and it's the corpus callosum that joins them. Remember, don't mix corpus callosum with corpus luteum. All right, we confuse those. They're two totally different structures. All right, and then the next part of the brain that you need to know is the hypothalamus. The hypothalamus is important. One of the things that also it does is it's part of your regulates some of the hormones, responses, thirst, sleep, hunger, those kind of things, tiredness. And then we have a look at the cerebellum, and the cerebellum's at the back of your brain, of your head, and that has to do with balance and equilibrium. And then we have this part that goes down from the hemispheres of the cerebrum and the cerebellum, and that is known, the lower part is known as the medulla oblongata, and the medulla oblongata basically joins the brain to the spinal cord. All right, then if we look at the peripheral nervous system, we can see from the central nervous system, so all the nerves, the blue nerves that are branching off, that is known as our peripheral nervous system. Okay, and then if we have a look, we've got a somatic nervous system. Remember, that is where I said where we're working, or it's the nervous system that carries out voluntary actions or functions. And then we have the autonomic nervous system that carries out functions automatically, so we don't actually have to think about it. Right, then another important aspect of the nervous system that you must know is the reflex arc. Now, what is very important to know is the difference between an arc and the action. Right, so the reflex arc is the actual pathway, and the reflex arc starts at the sensory receptor, and in this case you can see the receptor is in the skin, and then it goes via what we call a sensory neuron. Okay, so there's our sensory neuron, and our sensory neuron is going to carry the impulse into the spinal cord, which is part of our central nervous system, and there the sensory neuron is going to synapse with a connector neuron or an interneuron. Okay, so it's the one, inter means between. Okay, so that's the neuron then that's part of the central nervous system, and it's going to analyze and integrate the information that it has received via the sensory neuron, and then it's going to effect a response. Okay, because that's why you're getting the information there, is so that balance can be restored. So what happens then is the interneuron then is going to, so it synapses with the sensory neuron, and then it also synapses with the next neuron in the pathway called the motor neuron. Okay, and the motor neuron is the thing that brings about the action, movement. Remember, motor, movement, and usually it would be a muscle or a gland or an organ that's going to bring about whatever reaction that is required. All right, so that's the basic structure and function of the nervous system. Okay, sorry, there we have the effector. All right, then let's have a look at the three types of neurons. As I said, we have a look at them as far as in the context of how they function. So the first one is a motor neuron, and a motor neuron usually is a multipolar neuron. So it has several dendrites that go into or enter the cell body, and then it has an axon that leaves the cell body. Okay, so remember, this is the cell body. And then the direction of impulse is always from the dendrites. So from the dendrites into the cell body, and from the cell body out, okay, towards the terminal ends of the axon. And then remember, the axon always is going to synapse with an adjacent neuron, but with the dendrites, okay, because the direction of impulse is always from dendrites to cell body to axon. Okay, you'll never get two axons facing each other and expect the impulse to travel. It's not going to. All right, so the first one, as I said, is a motor neuron, and that is the neuron that is going to carry the impulse away from the central nervous system to the effector, to the muscle or the gland that is actually going to bring about a response. Then we have sensory neurons, and usually sensory neurons are unipolar or bipolar, and there's a receptor cell, okay, and that it would be in one of our sense organs, whether it be in our skin or in our eyes or in our ears, on our tongue, inside of our blood vessels. And what it does is it is sensitive to a specific stimulus. And what happens then, as soon as it senses that stimulus, it initiates an impulse, and that impulse then is carried via the dendrite into the cell body, and then out via the axon again to the central nervous system. All right, so that's going to so that is when a stimulus from the environment is converted into an impulse and carried to the central nervous system. Then our interneuron is usually also a multipolar neuron, and basically what it does is it is the connection between the sensory neuron and the motor neuron. So that is your controlling center, and what it does is it analyzes the information that comes in from the sensory organ, the sensory neuron, and then it effects whichever appropriate response. All right, so those are the three types of neurons. And remember, neurons are specific nerve cells, and neurons differ. They're different from nerves. Nerve is actually a bundle. So it's a bundle of neurons makes up a nerve. Okay, so don't confuse nerves and neurons. So if you think of an electrical cable, if you have a look at it's a thick rubber cable, but if you cut through it and you have a look inside, you'll find a whole lot of copper wires inside there, and it's those individual copper wires which would be the neuron, and then the bundle, when we bundle those neurons together, we form a nerve. Okay, all right, so let's have a look at some questions. This is a question from a prelim paper from Limpopo for this year. And what they have here, they say the table below represents the percentage of people affected by Alzheimer's disease, or they say AD, in different countries. Now remember, Alzheimer's is the degeneration of the nervous system. And so what's important here is they've given us a whole lot of information in that first. They say the table represents, and there's a independent and a dependent variable there in that statement. All right, but the first question, okay, so let's look at the table. Here we see the different countries. So they tell us there's various countries. We can see it's South Africa, Mozambique, Finland, United Kingdom, and Mexico. And then they give us the percentage of people with Alzheimer's disease. Okay, so it's a percentage. South Africa is 16.5%, Mozambique is 23.9%, Finland 55%, United Kingdom 43%, and Mexico only 3%. Right, the first question they ask, they say, explain why people with Alzheimer's disease should be under constant supervision and not allowed to walk alone. Okay, so remember when Alzheimer's occurs, it's the nervous system or the neurons start to degenerate. And so what happens is people then become confused. They often suffer from memory loss, and they can get lost. They become very confused, as we've said. They are unable to do simple chores and that kind of thing. They don't know who they are very often. They don't know who their family is. They forget. So this is why usually Alzheimer's patients are unable to function independently. So very often they need care. All right. The next question. Okay, so yeah, they get to the nitty-gritties, and we especially put this question in because they've asked us to draw a graph. And remember, in life sciences, both paper 1 and paper 2, there are certain aspects or certain questions that we can get easy marks. All right, and this is one of them. So we're not sure whether the graph will be in paper 1 or paper 2, but we need to be prepared for it to be in either of the papers. So here, and what is very important is straight away, we've already got 1 mark if we follow the instruction. Because in the question it says, draw a bar graph. And if you have a look at the criteria, have a look there, and how we mark the bar graph, it says, or the criteria, it says a bar graph is drawn, and I get 1 mark. Actually, I just want to, so there's 1 mark that you're going to get. So if we follow the instructions, and we draw a bar graph and we don't draw a line graph, so you need to know the difference between, and specifically a bar graph and a histogram, because that can become confusing. Remember, a bar graph has spaces between the bars, whereas a histogram doesn't have any spaces between the bars. Okay, so there already I've got 1 mark. And then we're going to draw the graph, and what's important then again is to have a look at the information in the table. So I'm just going to go back to the table, and there we can see there's percentage of people with Alzheimer's disease. So that's what we're going to record, how many people in the different countries. All right, so when I work out my scale on my y-axis, which is going to be my dependent variable, so the percentage of people with Alzheimer's, I can't use the numbers that are in the table, because then I'm not going to have, my graph is not going to be, it's all going to be a straight line or straight, there's not going to be, it's not going to be show the picture that it should. So I must have a look at the numbers in here, and I can see my lowest number is 3 and my highest number is 55. So I need to work out a scale that is going to include all of those. But I'm not going to use the exact, for my scale, I'm not going to use the exact numbers that are in the graph, in the table. All right. So I start at 0 and I'm going to go up to 60. So that will include 3 as well as 55. Okay, but I make them equal increments. So you can see on this graph here, we go up in increments of 10. It must be equal. All right. And if I do that, then I'm going to get, my scale will be correct. All right, so I'm going to then draw my y-axis like that, and the x-axis then is going to have the countries on it. So each country is going to be represented by a bar. And what is very important with that is that when we draw the bar graph, the width of the bars, so from this bar and that bar and that bar, must all be exactly the same. They must all be equal. So it's very easy if you just know. And the spaces between the bars, so that space and that space, that space and that space, they must all be equal as well. Okay, and then I'm definitely going to get my scale mark. All right, so let's work through this graph through the criteria. So first of all, we said we're going to get 1 mark if we draw the correct bar, the correct type of graph. Then if we have the correct caption, Now the caption is a heading, and the caption must always include both variables. So you can even put in your heading or your caption afterwards once you've drawn your graph, because then you know which variable is on which axis. And remember, the one on the x-axis is going to be the independent variable, and the one on the y-axis is going to be the dependent. All right, so I need to include both of those in my heading, my caption. So unfortunately, they didn't state it here in the question, so I go to the table, and the table tells me that they're illustrating the percentage of people with Alzheimer's disease in different countries. Okay, so then I've got both my variables. There's my percentage of people, and here my different countries. All right, and then in this case, they've told us it's percentage, but if they didn't say specifically, then I would have put in percentage, my units. I always need to indicate my units, okay, in a graph. All right, so if I've drawn the correct type of graph, I'm going to get a mark. If I've put in the caption with both variables in it, I'm going to get a mark. If I've got the correct labels, so I must label my X and Y axis correctly, I'm going to get another mark. If my scale is correct, and remember I always have equal increments on my Y axis, and then my spaces between the bars and the bar width must be equal on the X axis, I'm going to get a mark. And then I'm going to plot the information. So then from the table, I'm going to use those values and I plot them. And in this case, if I plot 1 to 4 correctly, then I'm going to get 1 mark. But if I plot all 5 of the bars correctly, then I'm going to get 2 marks. So actually, a graph is a very easy question. If you have practiced it and you know the difference between the graphs, it's very easy marks that you can get. So there's 6 marks there without too much difficulty. Right. Then here this question comes from the Eastern Cape Prelim Paper 1. Here they say this diagram shows a reflex arc. It's slightly different to the regular or the usual reflex arcs that we see. We don't normally see this part of the spinal cord. Okay, so don't let it confuse you. That is just the spinal cord. And there you can see peripheral nerves branching off the spinal cord. So here, but we're only going to have a look at this part here. All right, and there we can see our reflex arc very clearly. All right, so if we have a look, they say, the first question says, give the letter that represents the central nervous system. And if I have a look, A is labeled up there, but that is the spinal cord that they're showing us. And so it would be A. Right, then they give us a little scenario. They say a person stepped on a nail with his foot and immediately pulled it away. Okay, you've all experienced that. Now what they want us to do is they want us to describe the path of the reflex action. Okay, so the reflex action was pulling that foot away very quickly. Now they're asking you to describe the path that the impulse traveled along, so that from where the skin or the foot sensed the pain, okay, the painful stimulus, to when it actually withdrew the leg, the foot. Okay, so the reflex action from the moment he stepped on the nail until he pulled his foot away. And have a look, it's worth 5 marks. So I need to actually describe what happens along that pathway. Right, so let's have a look. The first thing, the receptors in the skin, as I said, there's pain receptors that are in our skin, and they receive the stimulus. Okay, and what they do is they then convert that stimulus into a nerve impulse. Please don't use the word message. They don't convert it into a message. It's converted into a nerve impulse. That nerve impulse is then carried via the sensory neuron. So the sensory neuron then is going to conduct or transmit that nerve impulse, and where does it take it to? It takes it to the central nervous system. So it transmits the nerve impulse to the interneuron, because remember we said that is the connector between the sensory neuron and the motor neuron. So the interneuron then receives the stimulus, the impulse in the spinal cord, and then it's going to transmit that impulse, because now your body wants to protect itself. So the thing to do is to pull the foot away. So now it must effect a response. So that is going to then transmit the impulse to the motor neuron, and the motor neuron is going to go to the muscles in the leg, the specific muscle, and the muscle is going to pull the leg away. So that would be your effector. Okay, so the muscle is going to contract, that will pull the foot away, and that is what has happened. So that is what actually happens in a reflex action. Okay. All right. Okay, now this question, and I know there's a lot of learners, and we often, or examiners often ask this kind of question. So they've said here, let's have a look again. In the diagram, A, B, C, and D represent different regions of the human nervous system. Then they say, in a car accident, different people obtained different nervous system injuries. So there were different parts of their nervous system that were damaged. So at the hospital, the doctor used a blunt object to gently scrape the soles of the feet of each patient to test for nerve damage. Write down the correct letter only. So you only have to give the letter. You don't need to name the part. For each statement below to indicate the damaged region of the nervous system. So the first one says the patient can feel. So when the doctor scrapes on his sole with that blunt instrument, he can feel it, but he can't pull his foot away. Okay, so he can feel the scraping, but he can't move his feet. So which part of the nervous system? So the sensory, he's still able to sense what's happening, but he can't move. So the sensory neuron would be working. So that part which we see as labeled B, that would still be working, but he can't move his feet, which would be D, which is your motor neuron. So the part that then would be damaged would be D. The next one, the patient cannot feel the scraping. So while the doctor's scraping, that person can't feel the scraping, but he can still move his feet. But he's not moving it necessarily in response. He just can't feel it. So that means that the sensory neuron, that sensory part of the nervous system, would be damaged. So that would be B. Then the third, for C, it says the patient can neither feel the scraping nor move his feet. All right, so if we have a look at this part here, C, we see that it is a nerve actually, that because the motor neuron and the sensory neuron are combined into that nerve. So the part that would be damaged because he can't feel and he can't move his foot. So he can't feel the scraping and he can't move his foot. So that would be part C that would be damaged. And then the patient cannot feel the scraping. He can't move his upper limbs, he can't move his lower limbs, he can't move anything. Okay, so that would mean that there would be damage here in the central nervous system. Okay, because there's no information going anywhere. So even higher up, it's not even just his leg, it's his upper limbs as well. So there's damage to the spinal cord. So therefore, it would be A. Okay, so you just got to have a look at what the scenario is, what the person can do, what he can't do, and if it's something to do with sensing the pain or sensing something, like this is just a bit of scraping on the bottom of the sole, then it's the sensory part of the nervous system that is damaged. If the person can feel but they can't react, then that means the motor part of the nervous system is damaged. And if it's a more general, a far greater loss of movement and feeling, then it would be the central nervous system. Okay. Right. And here's another question, a very good, very nice question. And here they've shown us an example of a neuron. So they've given us a diagram of neuron. They say neuron 1. And then if we have a look at neuron 2, we can see that neuron 2, the myelin sheath, which is a fatty layer that is wrapped around the axon, seems to be broken down. You can see it's damaged. Okay, so there's something wrong. All right, so let's have a look at the questions. So they say the diagrams represent a normal neuron and a neuron of an individual with a certain nervous disorder. Okay, so first of all, they ask us some very basic questions on the diagram, and you can see they've labeled different parts. So they say identify part A, and part A are those processes that go into the cell body. So they are then known as the dendrites. Then they want to know, state the function of part D, and we see part D is here, which would be the axon. So that's going to carry impulses away from the cell body, usually to the effector. All right, and then the third question, they say, identify the disorder of the nervous system shown in neuron 2. Now remember I said to you it's myelin sheath that is wrapped around the axon, and basically what myelin sheath does is it speeds up, it's an insulator, and it speeds up the reaction, the impulse, the movement of the impulse. So the impulse is going to jump from the gaps in the myelin sheath. So that means that the impulse moves very quickly. So now we can see this is damaged, and that is known as a multiple sclerosis, or we call it MS. And remember, it's easy to remember multiple sclerosis. It's because the myelin sheath, that's the myelin sheath that is damaged. So remember, you keep the MS has to do. So it's multiple sclerosis because the myelin sheath is damaged. All right, let's have a look at some more questions. Now, here they say, and I've highlighted parts of this question because I want you to be aware of this, because this is a very common way of questioning where they ask for one reason. So they say, give one reason for your answer to question 2.1.3. And remember, we said it was multiple sclerosis, or MS, was the disease. So one reason. Now, you can write down 5 or 6 different reasons if you want to, but the markers are only going to have a look at your very first point that you have written down. And that is because on the memo we write mark first one only. So it's very important that you are aware that when the examiner says give one reason or 2 factors or 3 characteristics or whatever, that we only give the number that they are asking, because they're going to have a look at the first one or the first 2 or first 3, however many the question's asking. Any after that, we're not going to look at. So when you are also answering and you're not so sure of your answer, make sure that the one that you leave there is the most correct one. Because that is the one the markers are going to look at. Sometimes we start answering the question and we're not so sure, but then as we write, then we become clearer. And then our answer, then the second point we write down is the one that actually is the correct one. In that case, cross that first one out. Okay, because they are only going to look at your first answer. All right, so that's why I've highlighted there. So it's also very important to follow instructions of the questions. And in that way, you are also able to maximize the amount of marks you're going to get. All right. So the one reason why it is MS or multiple sclerosis is that it's damage to the myelin sheath that surrounds the axon. Okay. Then the next question they ask us is explain the effect of the disorder mentioned. So what happens now is when it's broken like that, my impulse doesn't travel as nice and cleanly as it would normally along the axon. So it's going to slow down the transmission of the impulse. It's going to be much slower. which is then going to lead to slower responses. So I'm not going to be able to react as quickly as I need to always. All right. Okay, so now we're going to move on to the eye. We need to know the structure of the eye. So you need to know the different parts, the internal structure of the eye. So remember, we've got 3 layers of the eye, and the first layer is known as the sclera. Remember, it's white, tough fibrous tissue that makes that sclera, and it's quite tough. It doesn't stretch. So the fluid inside then exerts pressure on the outside of the eye, giving the eyeball shape. And it's very important that the eye is nice and round shaped so that we get clear images formed on the retina. All right, but remember, in our eye, in the retina, we have receptors or cells that are sensitive to light. So the white sclera can't extend all the way around the eye. The front part of the sclera becomes transparent, and that is to allow light to enter our eye, and that is known then as the cornea. The other thing that you will notice about the front part of the eye, the cornea, is that it bulges out. So it's not quite round. Okay, there's more of a bulge at the cornea. So your eyeball has this round bulge here. And remember, we said the more convex a structure, the more it refracts light. And that is why our cornea, first of all, it's transparent to allow the light to move through, and then it is also quite convex so that it can refract the light so that we get a clear image forming at the back of the eye on the retina. All right, so that's the outer layer. Then we have a middle layer, and the middle layer, I'm just trying to find it here, is the choroid. Okay, so that's this inner layer, and if you have a look at that, you can see there's lots of blood vessels. It's the part that nourishes the inner lining of the eye, so it provides it with nutrients. It also has pigment in it, so it's dark colored pigment, which then is going to absorb light. Okay, so once the light enters the eye and an image is formed, we don't want that light to reflect around inside the eye, because then we're going to have blurred vision. So that's the choroid, and the choroid, that middle layer, extends then in the front of the eye over here as the ciliary body. Now the ciliary body has a very important function in accommodation. So it's when we're focusing, either it's something far away or something close up. And then it further extends down into or in front of the lens, and that part is known as the iris. And the iris is a muscle, is a layer of muscle that is going to control the opening, because in between or in the middle there we've got the pupil, and it's the pupil that allows the light to enter into the back of the eye. Okay, so that's the iris. The iris is the colored part, and it is muscle, and it is able to contract and relax, and in that way it controls the amount of light that is going into our eye. Behind the iris over here, there's the other part of the iris, you can see this round structure here, and that is our lens, and that is the part that is going to either become more convex or less convex, and it's going to then allow us to focus close up or far away. And that is attached to the ciliary body by these ligaments that hold the lens in position. So, and then in front of the lens, between the cornea and the lens, we have fluid. It's quite liquidy, quite watery. It's known as aqueous humor. All right, and then behind the lens, and in most of, or making up most of the part inside of the eye, is the other more jelly-like vitreous humor. Okay, it's like a ball of gel. Okay, and we find that. Then if we have a look at the inner lining, and that is known as the retina, and the retina is the part that has our photoreceptors. So in there we have all the cells. They are called cones and rods, and they are then sensitive or they are sensitive to light. Right. So, and then if we have a look, if we look directly behind or straight, if the light passes straight into the eye, there's an area that's known as the fovea centralis, or the yellow spot, and that has the most or the highest concentration of cones, and cones are receptors that allow us to see color. Okay, so we're going to be able to see very clearly if the light strikes that area. We're also going to be able to see fine detail. Okay, so when we're trying to read fine print, then the light must strike on that area, because that's where we're going to be able to read it. And then just below that, we have another area here where our optic nerve, so all the little neurons from all the sensory neurons from the cones and the rods then accumulate into one big nerve known as the optic nerve. And that then leaves the eyeball at the back, but it's slightly low down. And that area there, where the optic nerve actually leaves the eyeball, there are no receptors. So we talk about that as our blind spot. Okay. All right, so those are the basic structures and the functions of the different parts of the eye. And then one of the important functions obviously is accommodation. And here we can see in this diagram, we can see that this is the lens over here, and the suspensory ligaments, and we can see they're quite tight. Okay, remember, suspensory ligaments can't contract or relax, but when the muscle contracts or relaxes, then it's going to, actually when it relaxes, it pulls those sensory ligaments tight. And when those ligaments are stretched or pulled tight, then it stretches out the lens. Okay, and so the lens then becomes less convex. And so if you have a look, if this is my lens in the middle, and I'm pulling further away, it means my lens is becoming thinner. I'm now able to focus objects that are far away. Right, so just remember that when you pull away and you make your lens thinner, okay, less convex, then I am able to see further away. When my lens becomes, so when the suspensory ligaments loosen, okay, and there's not so much tension on my lens becomes more convex, and so I'm now going to be able to see close up. Okay, so when we want to see close up, we need our lens to be more convex, so it must be quite fat and round like that. So it's more convex, which then increases the refractive power, which means that I'm going to be able to see close up. When my lens is less convex, so there's less, the refractive power decreases, I'm then going to be able to see further away. All right, so let's have a look, and it's very nice actually to compare these two in a table, because if you know the one side of the table, then it's very easy to remember, because it's usually the opposite. So if we have a look at when an object is less than 6 meters away, so it's close up, the ciliary muscles contract. Okay, so remember the muscles of the ciliary body. Okay, so they're going to contract. The sclera is pulled forward. The suspensory ligaments, so they become slackened, because so your ciliary muscle contracts, and then your suspensory ligaments become loose. And when they're loose, they're not pulling on the lens, so the lens now becomes quite nice and convex, becomes more convex, because the tension has decreased on the lens, and that increases the refractive power. Okay, so to try and remember this, we have this little mnemonic. So we say CC, which is ciliary muscles contract, suspensory ligaments slacken, that means they become loose, tension decreases on the lens, and the lens becomes more convex. And that means then I can focus close up. So when an object is more than 6 meters away, so now we're doing the opposite. We're having a look at further away. The ciliary muscles relax. The sclera is pulled backwards. Suspensory ligaments become tight or taut because they pull. The ciliary muscle then relaxes, so it pulls on those suspensory ligaments. And when it pulls them tight like that, the tension on the lens is going to increase. And then the lens becomes less convex. And so the refractive power decreases, and I'm able to then see further away. Okay, so we've got a mnemonic there as well for us to remember. So we're going to say ciliary muscle relax, CR. ST is my suspensory ligaments become tight, which means my tension increases on my lens, and my lens then becomes less convex. And then that allows a clear image to be formed on the retina. Okay, so remember, if you remember the one side, then it's very easy to remember the other side. Okay, for this. Right, let's have a look quickly at pupillary mechanism. And pupillary mechanism is the pupil of the eye that is able to either dilate or constricts because it regulates, or it's not actually the pupil that does it, it's the iris that causes the pupil to dilate or constrict, and that regulates the amount of light that is entering the eye. Remember, we don't want too much light entering the eye because it can damage the retina. The retina is a very delicate tissue layer. All right, so again, when we have bright light, so when there's very bright light, then our pupil is going to constrict because we want to limit the amount of light that's entering the eye. We want just the right amount of light in. We don't want too much light in. All right, so in bright light, now remember here we're talking about different muscle. We're talking about circular muscle. So it's important because if you have a look at the iris, the muscle is arranged in, it looks like circles. All right, so the fibers are arranged like that. Don't confuse circular muscle with ciliary muscle. Ciliary muscle has to do with accommodation. This circular muscle, so what happens is the circular muscle is going to contract. When it contracts, the fibers get shorter. All right, and that then is going to cause the pupil. The radial muscle is going to relax because we also have radial muscle. That's the muscle that is arranged like that. And that is going to relax. And what happens is your pupil is going to close down, or it's going to constrict, which then limits the amount of light that is entering the eye. When we are now in dim light, so there's not a lot of light. So now I'm not going to be able to see unless I allow all the light that there is into my eye. So what happens here in dim light, the circular muscle relaxes, and when it relaxes, the fibers get longer, and that means my pupil then dilates, it gets larger. All right, and my radial muscle then is going to contract, and that then is going to cause the pupil to dilate, which then allows more light into my eye so that I can see better. Okay. All right, so again, we've got a mnemonic, and we say in bright lights, we have circular muscle that contracts, radial muscle that relaxes, and the pupil then constricts, and less light enters the eye. All right, when we are in dim light, we say the circular muscle relaxes, the radial muscle contracts, the pupil dilates, and we have more light entering the eye. Okay, so these kind of mnemonics are quite easy to remember, to learn. And then when you get your paper and they ask you this, when you are able to write, not during your reading time, you can quickly write those mnemonics down so that when you get to the question and you answer it, you remember exactly what happens. Okay, now the differences between accommodation and pupillary mechanism Okay, so in accommodation, the stimulus is a change in the distance of the object. Okay, so whether it's further away or closer up, whereas in pupillary mechanism, it's the amount of light. So it's the light intensity that changes. The parts that are involved in accommodation are the lens, the suspensory ligaments, and the ciliary muscle. Whereas in pupillary mechanism, it's the pupil and the iris as well, and then specifically it's the radial muscle and the circular muscle that is going to then bring about the changes. The main changes that occur in accommodation is the shape of the lens. So it either becomes convex, more convex, or less convex. Whereas in pupillary mechanism, the pupil, the size of your pupil is going to change. Okay. And then what brings about the change? The shape of the lens is changed by the ciliary muscle that either contracts or relaxes, and that's either going to pull on the suspensory ligaments or it's going to release the tension on the suspensory ligaments, and that will bring about the change. Whereas in pupillary mechanism, it's the circular muscle, either when it contracts or it relaxes, it's going to bring about the change, as well as the circular muscle, as well as the radial muscle. Right. Okay, so here's a very good question, a very nice question. And if you look at the diagram, you first got to have a look to see exactly what that is. So if we have a look at the diagrams, we can see this would be the cornea. Here's the iris. This would be the lens. So M would be the lens. Okay, and there's your suspensory ligaments there. Okay, so already I must first have a look and see. Now, if I have a look at all four of the diagrams, A, B, C, and D, I can see that the lens is different in each one of them. The iris is also changed. Sometimes it looks smaller and other times it looks larger. So they're going to ask me, obviously, on that. All right, so let's have a look at the information. They say, the diagrams A, B, C, and D below show part of the eye under different conditions. The first question they say is name the process that involves the changes in the curvature of part M. So we already identified M as the lens. So we said the part of the process that changes the shape of the lens is known as accommodation. So let's have a look. Correct. Right, then question 2. Give the letters of 2 diagrams. So now only 2 of those, A, B, C, or D, that represent the eye of a person whose part M has a high refractive power. So when it is very convex, it's going to have a high refractive power. So we'll be able to bend the light the most. So I need to have a look and see which 2 diagrams. So if I look at A and I look at C, my lens is quite long and thin. Okay, so it wouldn't have a very high refractive power. So then I would eliminate those 2. So it's obviously going to be B and D. So let's see if we're correct. Yes. All right, let's have a look at the next question. Here they say, explain the changes that would occur in part N. So now they're asking us about part N. And part N we labeled as the iris. If a person was sitting in a well-lit room, so there's lots of light in the room, but at night, but there's a lot of light, and then suddenly all the lights go out. There's load shedding. Okay, and all the lights go out. Right, so what is going to happen to the part N? Okay, so now what we have to say, so we said the iris is going to limit the amount of light when there's bright light, but it's going to allow more light in when there's dim light. So let's have a look and see what they say. Okay, so the circular muscles relax and the radial muscles contract. So now remember there was bright light, so my pupil is small because I don't want to allow too much light in. So now what happens is my circular muscle must relax and the radial muscle is going to contract. And when that contracts, it's then going to open up or dilate the pupil to allow more light in. Okay, so the pupil size increases to allow some more light enters the eye. All right, then they say the next question, which diagram A, B, C, or D represents the eye of a person reading a book? Okay, so when I'm reading a book, like I'm doing now, in a room with bright light, so I'm looking up close. So that means if I'm looking up close, I need high refractive power, so my lens must be very convex. But there's lots of light. So I don't need, my pupil doesn't need to be wide. Okay, so I'm going to have a very convex lens, but I'm also going to have a narrower or a more constricted pupil. Okay, so let's have a look at which picture that would be. So if I have a look here, my pupil is too wide in diagram A, plus my lens is very narrow. If I look at B, my lens is nice and convex, as well as my pupil being quite narrow. If I look at C, my lens is too narrow. My pupil is correct, okay, but that's not correct. And then if I look at D, my lens is correct, but my pupil is too wide. So there's too much light coming in, so that one won't be correct. So therefore, the answer must be B. C, yes, okay. All right, and then the next one, they ask us, name the eye defect that results from the uneven curvature of part O. And remember we said part O is our cornea. And remember we said if it's not nice and even, then the eye defect that we suffer from is called astigmatism. Okay, and then the next question, again, they're asking, state one way. Okay, so they only want one. Don't give 3 or 4, just one way by which the defect named, so astigmatism, is corrected. And that would be to wear specific or specialized lenses. Okay, they're corrective lenses that is going to then improve the vision. Okay, or otherwise it could either be glasses lenses, or it could be contact lenses. All right, or they can use laser surgery where they shave off the top layers, the uneven layers, and possibly smooth out that cornea. All right. Okay, and there again they're going to only mark the first one. All right, so let's quickly go into the ear, structure of the ear. We know the ear consists of 3 parts. So we have the outer ear, we have the middle ear, and we have the inner ear. Now the outer ear consists of this part, which is the pinna, this canal here, and then the boundary between the outer ear and the middle ear is this membrane called the tympanic membrane, or the eardrum. Then our middle ear is a small region, and it's found here, this part here, and inside the middle ear are 3 very important little bones or ossicles, known as the malleus, incus, and the stapes. Also there is a tube that runs down into the back of our throat, and that is known as the eustachian tube. So that's the middle ear, and then the middle ear is connected to the inner ear over here by the stapes or the stirrup, where it's attached to a little membrane known as the oval window. And then the inner ear consists of also 3 parts. We have the semicircular canals here, we have the sacculus and utriculus down here, and then we have the cochlea there. And then we have nerves that are branch off from the inner ear. All right, so there's 3 parts to the ear. Okay, so very important. Let's just see. There we go. Okay, so remember the outer ear, the function of the outer ear, the pinna, and the shape of our pinna is that it channels all the sound waves into, so it captures the sound waves and channels them into the canal that then causes the little tympanic membrane to start vibrating. Then we have, from the middle ear, we have got the bones, and there you can see the tympanic membrane, so that's going to start vibrating as the sound waves enter the ear. and that the tympanic membrane is attached to the first little bone, the hammer or the malleus, and that then starts to vibrate. So as the tympanic membrane vibrates, it's causing the bone that is attached to it to vibrate, which then vibrates on the second bone, the incus or the anvil, and that then also, so the vibrations then are transferred from each of the bones till it reaches the stapes or the stirrup. And remember, the stirrup is then attached to the oval window. So our sound in the middle ear, the sound waves are then converted into vibrations, and the vibrations are carried through those little bones from the tympanic membrane to the oval window. Now, another very important function in the middle ear is amplification of sound. So because of the size of the tympanic membrane compared to that of the oval window, we have quite a lot of amplification. So in other words, the sound is amplified up to 20 times in the middle ear. Okay, because of the size difference. Right, then from the oval window, the vibrations are now converted. So now the oval window starts to vibrate, and in the inner ear, remember, it's a series of canals that have fluid in it. And as soon as they start vibrating, they're going to cause waves in the fluid. Okay, so the stirrup causes the membrane of the oval window to vibrate, and that sets up waves in the fluid. And then the organ of Corti is found in the cochlea, and as soon as the waves start to move, the cells that are in the organ of Corti are going to move up and down, and when they touch a membrane covering them, they're going to stimulate an impulse, and that impulse is going to then be carried to the cerebrum. Okay, where sound is, because sound is interpreted in the cerebrum. So that's basically the path of sound. Let's have a look. So we start with the sound waves that are captured by the pinna. They move through to the auditory canal, causing the tympanic membrane to start vibrating. So it starts to vibrate, and that then causes the ossicles in the middle ear also to vibrate. So now it's been converted from a sound wave to a vibration. If we have a look at the length of the sound waves, you can see here in the middle ear how much they have increased, because remember, that is where amplification occurs. And the ossicles then transfer the vibrations to the oval window, which is also a membrane, and behind that oval window is cochlear fluid. There's fluid in the inner ear, and that then is going to create pressure waves or waves in the liquid, and that then causes the organ of Corti, the cells there, to be stimulated, and as soon as they are stimulated, they're going to initiate a nerve impulse, and that nerve impulse goes via the auditory nerve, and remember, it goes to the cerebrum, because that is where the it's where you're going to interpret a sound. Okay, that's where hearing is going to take place, in the cerebrum. All right, now balance and equilibrium. And balance and equilibrium is basically keeps us that when we walk around, run around, when we're jumping, when we whatever, sitting down, standing up, we're not going to fall over. Okay, so balance and equilibrium is when our head, or the position or the speed and direction in which our head is traveling or moving, it stimulates receptors. So if we have sudden changes in speed and direction of our head, so say when you're running around on a soccer field or on a netball court, or you're running away from something, or you're running to catch the taxi or whatever, then that sudden change is going to cause the receptors in your semicircular canals to react or to be stimulated. Okay, so sudden changes in speed and direction causes the endolymph, because remember we said there was fluid in the inner ear, so there's endolymph as well in the semicircular canals, and they start to move. And in the ampullae, or the swollen ends of the semicircular canals, we find this structure. It's called a crista. Okay, so there's a whole lot of hair cells, and it's got a little jelly cap over it, and here is the endolymph. So this is the endolymph in here, and when that moves, what it does, so when the endolymph moves, it causes that jelly cap to move, and those little hair cells are bent, and then an impulse is triggered, and that then would be carried to the central nervous system. Okay, so those sudden changes, so the movement of the fluid then stimulates the cristae. Remember we said this sensory receptor is called a cristae, and it's found in the ampullae. Ampullae just means a swollen sort of part, base of the semicircular canal. So the semicircular canal it's like that, but then it has a swollen end that widens, and those little receptors will be found in the semicircular canal. All right, and then that allows us to, so as soon as the cristae are stimulated, then that allows me to maintain balance. So there's impulses that go to my muscles to make sure that while I'm running around, I'm not going to fall over. Okay, and then also the other receptors that we have in our ear are called the maculae, and maculae, they are found in the sacculus and the utriculus of the ear, of the vestibular apparatus, so that's that part there, and they are sensitive to gravity. So if I bend down like this and have a look at something on the ground, I'm not going to fall over. Okay, that's because those receptors then are stimulated, and they help my body to maintain balance. If I'm walking down the stairs, because my head is now changing, the direction is changing in response to gravity. If I jump up and I head at the ball, hopefully I'm going to come down, and you'll see most of the soccer players are able to land on their feet again. Okay, and that is because the maculae are stimulated. All right, so these impulses then are sent via the vestibular branch of the auditory nerve, and the auditory nerve now carries the impulses to the cerebellum. Okay, so it's not the cerebrum. Remember, hearing goes to the cerebrum because that is your area where sound is interpreted or hearing takes place, but your cerebellum is the part of the brain that allows you to maintain or restore balance. Okay. So the two ways in which the inner ear regulates balance, and again we've done a little bit of a mnemonic for you. So we use the word acids, and basically acids mean in the ampulla of the semicircular canals, the cristae are stimulated by direction and speed. Okay, so as soon as I'm changing the direction and I'm running or I'm walking slower or whatever, they are sensitive to that. So it's the ampullae in the semicircular canals which have the cristae, and they are sensitive to direction and speed, and they then maintain balance in that way. All right, whereas sump is the word that we use for our sacculus and utriculus, so the S and the U, they have maculae in them. Those are the receptors there, and they are sensitive to the position of your head in response or with regard to gravity. Okay, so when that changes, then they will then ensure that balance is maintained so that you don't actually fall over. Right, so let's have a look. Sorry, let me just get some help here.
I want you to ask yourself these 3 questions. Number 1, what do I want? Do I want to become an A student or do I want to become an average student? Number 2, I want you to ask yourself, how do you plan on getting there? Are you going to put more extra hours or are you going to just sleep? Number 3, who will be with you in this journey? Because it is important that we associate ourselves with students who have the same vision as us, who want to become a student, who want to put in the extra work, who want to become better at what they do. Unsuccessful people do not plan. They just let life happen to them. But successful people are planning on how they want their lives to be. And right now, you plan on how you want your results at the end of the year to be like.
Okay, sorry about that. Welcome back. Okay, so we said that there's 2 ways in which the inner ear regulates balance. So the first is the semicircular canals. They have cristae in them, which is the receptor, and that is receptive to direction and speed of the head. And whereas in the sacculus and utriculus, we find maculae, which are receptive or sensitive or pick up the change in the position of your head in relation to gravity. So both receptors then convert the stimulus into an impulse and send it via the auditory nerve to the cerebellum. And remember, so try and remember that cerebellum and balance, they sound similar. Okay, so you don't confuse and write cerebrum. It's cerebellum balance, which then sends an impulse to the affective muscles, and that's how I maintain balance. All right, a quick question on the ear. The diagram below represents a part of the human ear, and this was a question in last year's November exam, and I know that learners battled. They weren't able to answer all the questions very well. All right, so the first thing they ask us is identify part C. So we need to go and look, and we see here's part C. So we know that coiled snail shell is known as the cochlea. Okay, it's the cochlea. Then they say state one function of part D. If we have a look, here's part D. And remember, in the at the end of the tubes of the cochlea, there's another little membrane called the round window, and that round window acts like a valve, a pressure valve. So it releases excess pressure waves. It releases them so that you don't have ringing in your ears. So it's going to absorb the excess pressure waves or release the pressure wave from the inner ear, and it prevents the echoing. Then they say state one function of the receptors found in part C. The receptors found in part C are to convert the stimulus into an impulse. And then they say what happens, explain why a build-up. So we can imagine now there's earwax that builds up in part A, or builds up, and part A then obviously can't function, and that may result in temporary hearing loss. So we must explain why. So if we think of the sound waves going in here, and then there's a little bit of wax that is built up there, it's going to prevent the sound waves from reaching part A. And if part A doesn't vibrate, then the vibrations, the sound waves are not converted into vibrations, and therefore not into pressure waves, and therefore there will be temporary hearing loss. But let's have a look and see what they say. Okay, so part A, the tympanic membrane will not be able to vibrate, or will not be able to vibrate freely. So if there's no or there's less vibrations that are carried to the middle ear or the ossicles, then there will be a sound, or there will be hearing loss. Right. Now the next question, they say a grommet is a small device. So it's a little tube that they put into the tympanic membrane, into part A. They insert it, and it allows air to move in and out from the middle ear, and it prevents pressure buildup in the middle ear. So explain how the use of grommets in the treatment of middle ear infection prevents hearing loss. So what happens then is, as soon as we've got a middle ear infection, it causes fluid build up. So we get fluid to build up here, and then that causes pressure from inside, and that's going to affect the pressure against the eardrum, and it can actually cause it to rupture. So what happens then is that fluid build up can cause, and also causes the eustachian tube. And remember, the eustachian tube is going to equalize the pressure, and so it prevents that from happening. So what happens is, when I put in that little grommet, it's like a little tube, and then it allows the pressure that's from the middle ear to be released, and that then prevents the tympanic membrane from rupturing, and then doesn't affect hearing. All right. Okay, and then describe how the receptors in part B, so here we're having a look at the semicircular canals, are involved in maintaining balance when there are changes in the speed and direction and movement of the head. So we have a look. The cristae are stimulated, because remember we said it's the cristae that are found in the semicircular canals. They are going to convert the stimulus to a nerve impulse, and the impulses go via the auditory nerve to the cerebellum, and there that interprets the information and sends impulses to our skeletal muscle that maintains balance. All right, let's have a look at the endocrine system quickly and homeostasis. If we have a look at the endocrine system, we can see it's 34 marks, so it's a fifth of our paper, so it's also very important topics. And the endocrine system, there we need to know the basic hormones, the glands that produce the hormones and where their target organ, how they function. Then we also need to know about a negative feedback mechanism, and then homeostasis. And some of the things that some of the aspects or the functions of homeostasis, such as glucose regulation, CO2, water, and salts, we learnt in grade 11. So basically it's just a revision of your grade 11. And then we're also going to have a look at thermoregulation, which has to do with body temperature, how we regulate our body temperatures. All right, so if we have a look at this diagram, we can see all the main endocrine glands on here. And then, so what's important is you need to learn this diagram. You must know where the different glands are situated, and the hormones that they release. So first of all, the hypothalamus of the brain produces ADH, and its function is osmoregulation. Then the pituitary gland, its growth hormone, TSH, FSH, LH, prolactin. We're going to look at the thyroid gland, which produces thyroxine, the pancreas, which produces two hormones, insulin and glucagon, the adrenal glands, which produce adrenaline and aldosterone, and then in females, the ovaries, which is estrogen and progesterone, and the testes in males, which produces testosterone. Okay, so you need to know those hormones. Then a negative feedback mechanism is very simple. Okay, and a negative feedback mechanism doesn't mean that it's negative, has a negative effect on the body. It basically means that there's continual feedback happening. And the feedback mechanism always has three main components. The first one is the receptor. So something must pick up that there's an imbalance. And then what does it do with that information? That information must go to the controlling center, which is part 2. And that controlling center is going to say, okay, there's too much carbon dioxide in the blood. We need to maintain or restore the correct levels. So then that is going to then send the information, or it triggers an impulse that is then going to cause an effect to the effectors. And in this case, it would be your breathing muscles. All right. So if we have a look at homeostasis, we see that there's a sequence of 7 steps. And if you can learn these 7 steps, you can apply any of the examples they give you, whether it be blood glucose or carbon dioxide or water levels or thyroxine or whatever. You can apply it to these 7 steps. So the first thing is an imbalance is detected. So my receptors are going to pick up that there's an imbalance. There's either too much or there's too little. a control center is stimulated. So whichever control center of that specific function, then that control center responds and it sends an impulse to the target organs, and that target organ is going to respond and bring about a change. So it's either going to lower the levels if it's too high, or it's going to raise the levels if it's too low. And then in that way, balance is restored. Okay, so whether we look at the regulation of thyroxine levels in your body when they're high, if the thyroxine levels are very high, then that means the pituitary doesn't need to produce so much TSH, so it's going to produce less TSH, because remember TSH is the thyroid stimulating hormone, and if I've got lower levels of thyroid stimulating hormone, my thyroid is going to be stimulated less, and then it's going to produce and secrete less thyroxine, and that will bring the balance or the levels back to normal. And the same is if my thyroxine levels go too low. Okay, right, and again, for example, glucose. If my blood glucose is too high, then the information, so my glucose levels have increased, that information goes to the pancreas. The pancreas is then going to secrete insulin, and insulin then causes the liver and the muscle cells to convert the glucose. So it takes glucose out of the blood and it builds up a large molecule known as glycogen, and glycogen is stored starch in animals. Okay, so that reduces, so it's going to then decrease the levels of glucose in the blood. But now my blood glucose goes too low. Okay, so now that's the opposite. So we see the glucose levels decrease, that information gets back to the pancreas, and the pancreas is now going to stop secreting insulin and it's going to secrete glucagon. Actually, glucagon inhibits the secretion of insulin. So glucagon is the second hormone, and what glucagon does is it stimulates the conversion of glycogen back to glucose. So those large molecules, those storage molecules now are broken down into glucose molecules, and they can then go back into the bloodstream, and that then causes my blood glucose levels to increase. And so homeostasis basically is the maintaining of those levels within certain limits. We maintain the level. All right. Okay, so let's have a look here. This is a question from a prelim paper, and this is an investigation. And I know investigation questions are quite difficult, so that's why we're going to have a look at this. So an investigation there's a lots of information. Okay, and then there's questions based on it. So we need to read through quite a lot, but it's very important that you do. And they very often give us the aim, and they give us the way in which the investigation was carried out. They also might give us results. So we need to work through all of that. But in this one, they say an investigation was conducted to determine the influence of adrenaline on blood glucose levels of type 1 diabetics. Okay, so we know when somebody is diabetic, they do not produce enough insulin. So they have to take in insulin to regulate their blood glucose levels. Okay, so they want to know, what effect does adrenaline have? And remember, adrenaline is that hormone that allows us to react in extraordinary situations. And one of what the effect of adrenaline on blood glucose is it usually increases the blood glucose so that we have energy to get ourselves out of that extraordinary situation. Okay, so they say the investigation was conducted as follows. They used 150 female patients with type 1 diabetes mellitus. They were given the same amount of food and water at the same time for 15 days. Their blood glucose levels were measured. So they measured their blood glucose levels in the morning every 5th day. A solution with a low concentration of adrenaline was then injected, and then after 20 minutes, blood glucose concentration was measured again. The blood glucose levels before and after administering adrenaline were compared. So they recorded the results and they compared them. Okay, then they ask us, identify the dependent variable in this investigation. So I'm going to have a look at the aim. And if we have a look, we can always identify the aim where it says to determine, actually, the influence of adrenaline on blood glucose levels. Okay, so that's the aim. So we're going to go and look for our variables. Okay, so they say it's the influence of adrenaline, and so what is going to react? It's the blood glucose levels that are going to react. So our dependent variable should be the blood glucose levels. Then they say, the next question, they say, state 2 factors that should have been kept constant during the investigation. Okay, so we had a look there. They gave us a lot of things that were kept constant. And there, so they kept the same type of food. They said they were given the same amount of food, so the same type of food. The concentration of adrenaline should be the same. Can't give some more or more concentrated adrenaline than others. The person measuring the glucose concentrations must be the same person, that they measure it in the same way. The amount of adrenaline given to the patients or the participants must be the same. The age of the patients should all be the same, because possibly at different ages the metabolism would be different. The measuring tools that they used must be the same. The body mass of the patient should be the same. And any 2 of those you can give. All right, so we have a look at what, and remember, our factors that are kept constant are those that could affect the results if we don't keep them constant. And they then would going to affect the outcome of the investigation if they're not kept constant. All right, then the next question, they say, explain why the blood glucose levels were measured before injecting adrenaline on the body. So what they wanted to see was the effect of adrenaline on the blood glucose level. So if they don't measure the blood glucose before, they're not going to be able to see if there was any effect. So they need to measure before, so that they can see what it was, and then see whether there was a change. And so this measurement then becomes the control or the baseline, okay, the comparison, so we can see, did it have an effect, did it increase it or did it decrease it, but there was actually a change that happened. All right, and then the next one, they say, give a reason why the investigation may be reliable. And remember, reliability has to do with the size of the sample or the amount of times we carry out the investigation. We might repeat it. We might do a whole lot of measurements and then work out an average. But here, specifically, they told us there were 150 female patients that they used. So, okay, sorry. They used 150 patients, and the blood glucose levels were measured every 5th day. Okay, so this is why we can say the results are reliable. All right, then thermoregulation. Remember, that is a function of the skin. And in hot conditions, what's going to happen is the blood vessels just below the surface of the skin are going to dilate, and we call that vasodilation. So I just want to write it here. That's vasodilation. So the blood vessels dilate, and when they dilate, there's more blood that is able to flow through those blood vessels. And what they do is then they're going to give off heat. So the more blood that flows, the more heat that is going to be lost, either through radiation or through evaporation. It's also going to then stimulate your sweat glands, so they're going to become more active and they're going to produce more sweat, which means that the body is going to cool down. In cold conditions, our blood vessels are going to constrict, so we have vasoconstriction. So then the blood vessels get narrower, and when they are narrower, there's less blood that flows through those blood vessels. So we have less heat being given off or lost. Also, when we have less blood, we have less active sweat glands and therefore less sweat being produced. Okay, so have a look quickly at a question. They say diagrams 1, 2, 3, and 4 show part of a homeostatic process in humans. Identify the homeostatic process represented. So if we have a look here, we've got the brain, and then we can see here diagram 1, there's blood vessels under the surface of the skin, as well as in diagram 2. And in diagram 3 and 4, we can see there's sweat glands. And in diagram 3, there looks like there's no sweat, but in diagram 4, there looks like there's sweat. Okay, so the process then would be thermoregulation or temperature regulation. Name the process represented in diagram 1. So there we can see our blood vessels are quite narrow, they're quite thin. So that means they have constricted. So, oopsie, sorry. That means that there's vasoconstriction that has taken place, whereas if we look at the same blood vessels in diagram 2, we see that they are much wider. So that means that there is vasodilation that has taken place. Then 1.43, they say give the letter and the name. So now I'm going to give 2 answers, and you can see there the mark allocation is 2 marks each. So that the part that controls the homeostatic process represented in the diagram, and that's why we now need to go and have a look at the structure, the brain. Then we can see which part is the homeostatic control, and remember we must give both the letter and the name. Okay, so we can see there that is B, the hypothalamus, and then one that is less active on a cold day is going to be E, the sweat glands. Okay, so on a cold day, we're going to sweat less, so it would be structure E that would be less active. And then state the importance of the process taking place in diagram 4. Okay, so that would be sweating. So the significance is that it cools the body down, or heat is lost through evaporation, radiation, and sweat. Right, I think we've run out of time, so I'm going to move on. I want to go to the end and just talk a little bit about the exam. Fetals. Okay, so what I want to say for tomorrow is that you need to be prepared. Okay, so remember, study hard. Hard work always pays off. Use past papers. That's the best way to prepare yourselves for the exam tomorrow. Make sure you sleep enough tonight, and that you get to school on time. You don't arrive late. You're not locked out of the exam venue, which then is just going to cause a lot of nerves. And make sure you have some breakfast before, if you are able to eat, and then get to school on time. There are also on this presentation, there are links of videos that we have used in the past that you can also use to prepare for this session. And then remember, very important, the things to take into the exam room with you is you must have a pen and a pencil and have a backup. All right, have 2 pens or whatever in case one pen runs out. All right, then also you must have an eraser, because if they ask you to draw a diagram or they ask you to draw a graph, you're going to, and you know, if you make a mistake, you want to be able to erase your mistakes. Then also a ruler, very important. If I'm drawing a graph, I need to measure that width of my bars, the width between my bars. I need to make sure my scale is correct, whatever. So I must have a ruler. I also must have a compass in case they ask us to draw a pie chart. If they ask us that, then I'm able to draw a circle nice and round. And then also the protractor to be able to work out the sizes of the angles in that pie chart, so the angles of the different segments. I must have a calculator, and then I'm set to write the exam. Right, and then also very important is believe. I just want to leave you with this quote. It says, believe in you, because your voice is important, because your dreams matter, because there's only one you, and because you are worth it. So believe in yourself. We believe in you, and I wish you luck. But luck isn't all that it takes. It takes a little bit of hard work as well, so you need to study. And I also just want to say a special, or just mention my Tshwane South Diamonds out there, and wish you all luck, and hope you do well in tomorrow's exam. Thank you.